Welcome everyone to Flagship Studios. The title of this particular section is Approaching Rare Disease as Common and Common Disease with Precision. So I'm going to provide a little bit of context and then I will actually ask each of our panelists to introduce themselves and to give a brief introduction. So the theme here really is there have been a tremendous number of advances in new technologies, whether it's human genetics, single cell sequencing, and this has allowed us to define patient populations in very unique ways. And because of this leads to new approaches to therapeutic intervention. So for example, for ultra rare and rare diseases, we can now define patients not based upon a clinical phenotype or an individual mutation, but based upon shared biology and shared pathways. The flip side of this is that common diseases we can actually approach with a different level of precision that begins to resemble rare diseases. And so how do we think about these concepts and how does that lead to new medicines that are going to help the lives of patients? So we'll do quick introductions. I'll start with myself and then we'll just go down the line. So I'm Robert Plenge. I'm at Bristol Myers Squibb and I lead the research organization. Jason.
I'm Jason Park. I'm the CEO of Empress Therapeutics. And do you want me to describe what Empress does?
Yeah, just say a little bit more about Empress. So maybe what we can do is for each of you, a little bit more about your company and maybe why you're excited, just briefly about this particular topic.
Sure. So I'm the CEO of Empress Therapeutics and a partner at Flagship Pioneering. And what Empress is doing is bring genetics to chemistry. And we know that nature has been using microbes for over 3 billion years to make experiments between chemistry and biology. And what we believe is some of the most valuable chemistry is actually encoded into microbial genes that can be found inside each and every person. And what we've done is built a reverse translational platform to look at clinical data, clinical observations, and extract compounds that are encoded in microbial DNA. And why I'm excited about this topic is I think there's a lot of talk about data and genetics and computation, and the most important code is probably the code of life, and that's an underlying theme for all these companies.
All right, I think that's going to be a theme that we get to again and again today, and I like the idea of making these ideas actionable. All right, Michelle.
All right, so I'm Michelle Werner, and I'm the CEO of Alltrna. I'm also a CEO-Partner at Flagship Pioneering, and Alltrna is a phenomenal company that's focused on tRNA or transfer RNA, and it's all about tRNA, hence the name Alltrna. And what we're doing is engineering tRNAs to read through nonsense mutations, at least in the first instance. And one of the things that's so exciting about what we can do, which is very different from different modalities, is that we can look at many diseases at a time versus a disease-by-disease approach, which is really the approach that has to be done with all different technologies. So one of the things, that I'm so excited about this topic is because I think this really allows us this, different way of really bringing innovations to patients who otherwise wouldn't necessarily be able to do it. When we think about making diseases more common, we actually can be able to bring forward technologies and medicines for patient populations much more broadly than we could before.
So it's great. So you're starting to see the themes of expanding patient populations from in this case rare to more common, but we're going to come back to that. All right, Chris.
Yeah. So I'm Chris Austin. I'm the CEO of Vesalius Therapeutics and a CEO-Partner at Flagship. I'm a physician and a geneticist by training. Vesalius is a company that has developed, we think, for the first time, a truly integrated platform to do precision drug discovery for non-oncologic common diseases. And the reason I'm so excited about this, and we'll talk about what that means, is really comes from my training as a physician. When I was in training 30 years ago, and even up until now, physicians and patients are well aware that the clinical heuristics that we use, that archaic nosology that we use to diagnose patients, though it's very useful for clinical care, mixes groups of people with very different clinical syndromes, different progression rates, different responses to drugs, et cetera. And I think we've all hoped the genetics would be the key to unlock that complexity. And we've seen the remarkable things that approach can take in cancer and then in rare diseases. But it's only been within the last few years that the technologies and the compute power have allowed us to address the big problem, which is the 90% of people who suffer from polygenic common diseases. And that's why I'm so excited about this as a physician, because this, I think, finally allows us to do what I've been dreaming about doing for, dare I say, 30 years.
Yeah, so that's great. So you're starting to hear the themes. Technology underpins everything, but in each instance, there's a slightly different variation on this theme. And Chris's case is going from common to rare through precision definitions. With Michelle, actually the opposite, at beginning to expand the list of ultra-rare diseases into more common categories. So there's a theme that you've all touched upon, but what I'd like to do now is expand on the question of, why now? Chris, to your point, people have been talking about this for a long time. And I think this is something that happens a lot in our industry, where people criticize us for saying, you've been talking about this for a while. So why now? Why now? I don't know. Who wants to go first?
Well, I'll reflect back 20 years ago, almost 21 years ago, and Robert, you'll know what I'm talking about, that when the Human Genome Project was finished, there was an expectation that all diseases would disappear within 2 weeks. And actually, I had the unfortunate position to be at the Genome Institute and responsible for translating the genome to biology and health. And so, we have been talking about it for a long time. And as in most things, we tend to underestimate or overestimate the effect in the short term and underestimate it in the long term. And I think we're now at the point where we understand the complexity of the genome as its own organism, if you really think about it. All these interacting pieces that are affecting each other. We had this beads on a string idea that was just turned out to be entirely wrong. And we now understand that. And so it's in my mind, it's not one thing. I mean, if you look at what Vesalius is trying to do, it really is looking at the things you have to know in order to develop a good drug. That is, what is the patient population? What is the mechanism in that patient population? What is the target within that mechanism that's going to revert the phenotype? And how do you model it in a way that's predictive for clinical response? So it's not one thing, it's four things. So the Vesalius platform is an integrated platform, but it's based on the knowledge and the advances within human genetics, within large clinical data sets, large phenotypic data sets, the kinds, Robert, that you mentioned, the multi-omic kind of data sets that one can generate on human cells now, the understanding of gene circuits as units of function, and the ability to model these at scale in human systems, not in the 293Ts or the COS cells that we all grew up with, but in human-based systems, which are the target organism. I mean, something we've dreamed about for years. It's now possible. This is incredible. And that's why I think you're seeing this sort of giddiness in the panel here, because I think it's real this time.
I think it's true. I mean, in 2000 there was a single genome that had been sequenced. It was an amazing accomplishment, but now we have tens of millions of genotypes that have been individuals that have been sequenced. We've linked that to clinical data and also a multi-omic data sets that get to single cell precision. All right, who next?
Well, in the context of Empress, actually, we're in San Francisco, so maybe I'll make an analogy to the tech world where, we're seeing some tremendous discontinuous advances that really started with simple ideas. People have been thinking about, for example, having machines talk for ages. And what enabled that, why now, to your point, is the idea of compute power plus data, plus the ability to iterate and learn and feed that back into the platform. So to use some themes that you guys referenced at Empress, what we've done is we now know that, we've long known that important compounds are synthesized in nature and that you can find the clues for that synthesis in DNA. But what's changed, what's different, is over the last few years, really just the last few, there have been multiple orders of magnitude, similar to what you guys were just describing, multiple orders of magnitude difference or increases in the clinical data sets that have this genomic data that we can now go in, compare healthy and disease, very similar to what you're describing, and pull out insights as to what's really important here, what compounds, what mechanisms.
Yeah, so we've got the ability to scale the number of genomes, the computational power to be able to analyze all of this data. Michelle, what else?
So what I would say is, I think that if we look back and reflect back on the last 20 years, we can all agree that we've made a tremendous amount of progress in the numbers of therapeutic advancements for patients across different diseases. But when you actually look at the numbers of diseases that are being addressed by those advancements, in the grand scheme of the total number of diseases, it's actually quite few, right? So few diseases are getting the innovations. Many diseases are not. If we just look at the rare genetic disease space, there are 6,000 to 10,000 different rare genetic diseases, of which only 5% have an approved medicine. That's a shockingly low number. And so even when you think about the sort of unmet medical need for the patients that are in the 95% of diseases that don't have an approved medicine, it's quite substantial. So when we listen to health authorities like the FDA and the EMA and the MHRA talk about how are we going to solve the problem of genetic diseases, we can't take the same conventional approach of a disease by disease by disease by disease strategy, which is how we've been doing it. Whether it's mRNA or gene editing or you name it, right? That's the approach that we've been taking. So the real call to action, I think, is how do we tackle this 6,000 to 10,000 different diseases, many diseases at a time, because it'll take too many lifetimes to be able to really have an impact on patients. And that's how we think about the world at Alltrna, and where I think tRNAs can be uniquely positioned to be able to address that. But it's something that I think is biology is dictating it, but we're also hearing about that need both from health authorities and patient populations, including myself as a caregiver for a patient in the rare disease community.
Yeah, so there's a burning platform. So if I were just to summarize this, we have the data, we have the compute power platform, but most importantly, we have the burning platform, and that patients really need these medicines. And I think without all of these combined, I think we're not able to accomplish what we want to accomplish as an industry. Okay, so I'm going to change.
Can I just make one comment on that, which I think is Michelle's absolutely right. And actually, I did a, Michelle, you know this, I did a calculation a few years ago. How long would it take before every rare genetic disease has a treatment at the current rate? The answer is 2,000 years.
Yeah.
I can't wait that long.
I know.
So if anything speaks to the need for multiplexing, this tells us. But the other thing is that in common diseases, though all of us love to talk about the very real advances that have happened in particular diseases, if you talk to patients, they're extreme, and caregivers and physicians, they're really frustrated because, and I can tell you from a physician perspective, 90% of the diseases that I trained to treat 30 years ago, I could go back into the clinic today and the treatments are the same, despite all of the science. And so I think we have to realize, on the one hand, the need is huge, but what that also means is that the market opportunity is incredibly large. It's 20 times the current market. Because 95% is not treated, and that's so from a humanitarian perspective, a company building perspective, and a financial perspective, the opportunity here is just unprecedented.
Yeah, no, I agree with that. So there's a framework that I like to use, and what I'll do is the next section we'll kind of march through this framework. But at a high level, the framework is you start with a deep understanding of human biology, and I refer to that as causal human biology. But that's not enough. You have to be able to match a therapeutic modality to a molecular mechanism of action. And then the third step is to have a very clear path to clinical proof of concept to know if your drug is working. And I think if we can do all three of these, this will lead to increase in probability of success in the clinic, which ultimately will improve R&D productivity. So if we go through each one of these, I'm going to ask, we'll kind of divide it up among the three of you, but we'll start with causality and human. And Jason, I think I'll start with you, and then Chris, maybe go to you, and then Michelle, if you want to add something. But how do you think about not just human biology, but causal human biology?
Yes, I love that question. Well, one way to think about causality, actually, you've spoken about this previously, is nature has run all these experiments. And we have clinical outcomes, we have clinical phenotypes, and you can associate them back to that underlying theme of the genetic code. We can look at natural variations in the human genome, understand how that causes, and it's really just hypothesis generation that you then go test in the clinic. The way we've thought about it is there's actually all these other genes that are inside of us. There's 100 million plus other non-human genes that we haven't thought of as part of our human genome, but nonetheless, they're actually conserved and they're really important for health. And how we've done that is we've looked across thousands of people. We've actually looked across other species as well. We've asked the question of what's conserved, what's important, and we can apply the same techniques for causality with the human genome in terms of copy numbers, abundance, enrichment, depletion, et cetera, and ask, what about these microbial genes that encode chemistries might be causally linked to clinical outcomes that we started with? Again, we started with clinical data. So the way we prove causality is we start with the clinical data, we make an observation, we go back to genes that encode chemistries, and then just like any other drug program, you have to prove the causality in the lab at the end of the day.
I'm going to push you on that just a little bit, if that's okay, because kind of my training, and Chris and I are kind of aligned on this, I think, around germline genetics occurs prior to disease onset. But I think a challenge with microbial genetics is it can kind of come and go, and it's not necessarily present prior to disease onset. So it creates a little bit more complexity, I believe, for microbial genomes. So you mentioned doing experiments in the lab, or are there other things that you could do in terms of longitudinally profiling patients and seeing how changes in the microbial genome can actually lead to patient outcomes? So maybe just dig a little bit more into causality for microbial genomes.
Well, there are other data sets that you can look at that you alluded to, which are longitudinal or interventional. Yep. There may be interventional data sets where you've seen patients respond to therapies and changes in the microbial genetics that are associated with that. And again, at the end of the day, it's just hypotheses that you go back in, you say, here's the compound, here's the mechanism. Does it make sense in the context of the clinical outcome?
Yeah. So I think it's a very powerful statement because it's the experiments in humans. It's randomized and that lots of people have actually had the exposure. But then because you can follow over time and often with interventions, I think that's a good way to establish causality. So I agree. All right. Yeah. Thoughts, Chris?
Yeah.
Causal?
Yeah. Just a couple things. First of all, I think from our perspective, rooting everything we do in germline human genetics is the sort of touchstone that we have for establishing causality. But what we've been able to do there, just to drill down on what we've managed to show, is what we believe is a sort of genetic equivalent of Koch's postulates. If you remember that whole business with infectious diseases, that is, we can go from characterizing large clinical data sets, all of whom have the same diagnosis, into identifying more specific clinical syndromes, which then we can use the genetics in each of those populations. The reduced complexity of the phenotype allows us to identify polygenic gene circuits that are operative in each of those, identify a target which is abnormal, which we think is going to revert the phenotype. But then we can model that in human-based cellular systems from the people who had the phenotype in the first place. And if we can demonstrate, which we can do, that those human-based systems have a sine qua non of the disease, something which we know is causative of the disease, and our compounds can reverse it, then we've completed the whole chain of causality in a way that the whole shots on goal approach, if you remember that back in pharma, just was not able to do. And that's a very exciting development. I think the other thing that though I continue to be jealous of my Mendelian colleagues, I think the underappreciated advantage of working in common diseases is that, of course, common alleles appear in the so-called undiseased population, in the general population. What that means is that one can use normal cells and normal perturbations of normal cells in people who have those supposedly normal people or asymptomatic people who have extremes of genetic risk. We can identify through phenotypes that we can identify in those cellular populations particular phenotypes, which we know are linked to the disease via the genetics. And when we can revert those cellular phenotypes, that's also a very convincing argument that these are important in disease. And then we can do really remarkable things from a technological perspective. We haven't mentioned doing genome-wide association studies in a phenotype in a dish. So one can do genome-wide association studies of 1,000 people cells within a dish. And so use those genetic associations to establish causality as well. And we're doing all of those things. And none of those things were possible, even 5 years ago, much less putting them all together.
Yeah, that's great. Michelle, anything
The
to
one
add?
brief thing that I would just add is, I focus on genetic diseases, really. And we know that it's mutations that cause these genetic diseases. But we often say, okay, you've got this disease because you've got this dysfunction in this protein or this mutation in this particular gene. But that's not good enough. What we really need to be getting to is what type of mutation and what specifically, right, is changed in that code in order to really, truly understand the causality.
Yeah.
And so really having that, genomic sequencing, I think, is going to become more and more important as these precision medicines come forward.
So that's it'll be a nice transition here. So one of the things that I would challenge the audience is anytime anyone presents a therapeutic hypothesis, always ask the question, what is the causal human biology to support the therapeutic hypothesis? And my guess is, I don't know, increasingly it's becoming more common, but often there's not a causal link. And if you do not see a causal link, you should be very suspicious about the therapeutic hypothesis. Okay, but you raised a very good question, which is, it's not just understanding causality. You have to get to mechanism. And the mechanism is not just a biological mechanism, but then how do you match that to a therapeutic modality? Because that's what we do. We develop medicines, we intervene. So tell us a little bit about the proliferation of modalities, Michelle, and how you think about matching modality to mechanism.
Yeah, so I think that we're really in a golden age of sort of the emergence of genetic medicines, which I think is a truly exciting time. We've got mRNAs, we've got siRNAs, we now have our very first CRISPR therapeutic that's approved. And I think that's fantastic. We're talking about complex diseases that, all of these different ways are trying to tackle that challenge of genetic diseases in a fundamentally different way. So what we're doing at Alltrna, really thinking about how we connect modality and mechanism, we really truly understand the genetics behind each specific mutation. As I mentioned, we're looking at engineering tRNAs for nonsense mutations, at least in the first instance. Nonsense mutation is, occurs 10% of the time across all genetic diseases. So roughly 30 million people around the world have a nonsense mutation. And this is when you have, instead of a code for an amino acid, you have a code for a premature stop instead. And our engineered tRNAs can, precisely bind to these premature termination codons. So instead of having a disruption in that protein translation process, we actually can restore that translation process for a full-length functional protein. We think very differently about patients because it doesn't matter, what the disease is that they have. We really think about, what is that specific mutation, and do we see those mutations commonly across different diseases? And we do, actually. We see the exact same premature termination codon mutations across thousands of different diseases. So for us, our approach is really to cluster those patients instead of by that sort of classical phenotypic definition of a patient by a disease, but rather by those shared mutations that are the same across many different diseases, where one engineered tRNA may be able to address those mutations across those several hundreds different of diseases.
So hold that thought.
Okay.
Actually, I shouldn't hold you, because so we're going to come back to that, because I think that concept is so critical in going from ultra rare to common, and the ability to define not just a phenotype, but a molecular phenotype that can be expanded. So we're gonna come back to that. But the way I like to think about it is I think the number that I have in my head, and maybe you all have a different number, but 20 years ago, 90% of the approved drugs were small molecule therapies. If you look at it today, it's far more diverse. And then we have all of these emerging therapeutic modalities that aren't yet approved, but that we're really excited about. All right, so Jason, you have a particularly unique approach, I think, to identifying molecules for therapeutic intervention. So tell us a little bit about how you think about matching therapeutic modality to mechanism.
Well, we think of ourselves as a chemistry forward. It's a small molecule platform at the end of the day. And one way that we thought about it is there are many modalities. I mean, Michelle just spoke very eloquently about the benefits of tRNA and a number of diseases that you can address with some modalities in ways that you just probably can't with small molecules. There are some problems that you can't solve with chemistry. Nonetheless, to your point, I mean, the industry actually started with chemistry. And it still remains true that of modalities, it's small molecule drugs are the ones that are most consistently easily taken orally. They can be administered through various routes of administration. They're relatively cheap and easy to manufacture and distribute. They're still the only modality that regularly gets inside of cells and throughout any part of the body. So there are many advantages to small molecule drugs. And we think that there are a number of diseases, and we've looked at over a dozen indications where we think we might be able to make something completely novel here, bring novel molecules, novel targets, and novel mechanisms. So there are many diseases that I think you can still address.
Yeah. So let me say it a slightly different way. And I think that I understand it, but maybe I'm getting it wrong. So we have this chemical diversity that many of our small molecules have originated from in terms of approved medicines. But you're saying that nature has done the experiment to create a diverse chemical library that we have yet to explore, and you're looking at that chemical library that nature has created in the setting of humans and the germline genotype together with microbial genotype, and it allows us to explore not just chemical diversity but phenotypic diversity. Is that,
Both,
is that kind of it?
Just to put a number to your comment right there. One of the things that we've done is we've looked. There's a couple billion biosynthetic genes that encode instructions for how to make chemistry. If you look at the number of mutations and then the amount of time, the number is staggering. I think it's somewhere in the 10 to the 20th or 10 to the 24th number of potential compounds that have been synthesized just by the biosynthetic enzymes within our own body. So yes, absolutely, there's a tremendous amount of chemical diversity. And I think as an industry, we've only scratched the surface of what chemistry can do.
And we're also proud of our screening libraries in industry, where we can say we've screened 1 million chemical compounds, and you're like, yeah, come on, please. It's like a couple of billion, trillion, and beyond that.
Well, nature's
billions,
been out
billions.
a whole lot longer.
Yeah. All right, Chris, anything to add to the matching?
Yeah, all I would say is that to us, this proliferation of modalities is, we feel like a kid in a candy shop, because of course what we identify are points of intervention in particular pathogenic circuits in particular populations, and we can model them in those patient populations. But in the old days, we would have had to look specifically just for the small molecule targets, right? Just for the GPCRs, kinases, nuclear hormone receptors, channels. And now we can when we identify a novel, the correct intervention point in a circuit which moves the biomarker in an optimal way, then we can choose that target based on the biology. Yeah. Based on the biology, not on the fact that we have this limited tool that has to do everything. Yeah. And that, again, it's completely different than it was even a few years ago.
Yeah, no, I agree with that. Okay, so we define causal human biology. We now have matched a therapeutic modality to a molecular mechanism of action. We need to take it into patients. Yeah. So Chris, I'm going to start with you, and then I'm going to go to Michelle, because we're going to play off the theme of, rare to common, common to rare. Yeah. So how do you think about path to clinical proof of concept, defining those patient populations for your indications of interest?
Yeah, so it's an absolutely critical question, and I think the way I would start is to say that in our platform, we start with what the patient actually has. And what that allows us to do is to find much more comprehensively and precisely what are the things that change how the patient feels, functions, or survives, we start with that. And that, of course, is what MTA wants you to demonstrate with your intervention at the end of the day. So we're starting out with a much more precisely, if I may use the term, comprehensive characterization of what a patient has. We then are linking that to their genetics and to a biomarker that comes out of that circuit that we identified through the genetics. So now we have a precise and comprehensive and precise clinical phenotype, which, by the way, is like in Michelle's case, is not going to be just specific to the disease that we identify it in. It's very clear that these mechanisms and these clinical feature types, as we call them, are going to cross traditional nosologic categories. So, for instance, one of our earlier programs is in Parkinson's disease, but there's absolutely no doubt that the biology that we're studying is also operative in what we traditionally call Alzheimer's disease, ALS, frontotemporal dementia, et cetera. So we're having to do the same kind of thing that Michelle is doing. The challenge is, how do you identify those people? Because in common diseases, and Robert, you know this well, common diseases are not deterministic. The genetics is not deterministic, it's probabilistic. And so the question that we ask is, how robust does the clinical findings have to be? Can we base the patient selection just on clinical characteristics? Probably not. What role do the polygenic risk scores play in choosing those patients? In our opinion, my opinion, for most diseases, it's not going to be terribly useful by itself. The biomarkers that come out of those polygenic risk scores and the circuits that are identified, very, very useful. And so what we're looking at is from the very beginning, clinical phenotype, cellular phenotype, genetics, biomarker, and that is the package that we take forward to the KOLs and the regulators. Now, I won't sugarcoat this. When you go to KOLs who have been used to, were trained in, and have been used to diagnosing and treating a disease characterized by such and such over the last 20 years, and the regulators too, are pretty particular that you're not just making up something new for your own convenience, and that's of course entirely fair. But I think what we're looking for is not a division, Group A versus Group B, and they're completely different. We're never going to get there. This is going to be, can we improve the response? Can we improve the response rate from what it typically is, 10, 20, 30%, to 50% or 60%? If we can do that, that is a huge win for patients and regulators.
And maybe just to be very concrete, so you take a disease, you mentioned Parkinson's disease, and you have a clinical definition. Is what you're proposing to subset, is it a DNA biomarker? Is it a protein biomarker? Like, what is it that you're going to use to define that patient subset? And then maybe you answer that quickly, then I'm going to go to Michelle, and then I'm going to build on that question when I get back to the regulatory piece in a few minutes, but maybe be as precise as possible with how you define it.
Just in our current programs, they are biochemical analytes that we can measure both in the patient-derived systems and in people. And they come out, and they are the biomarker, they are the product of the biochemical or signaling circuit. They're biochemical circuits.
Are they proteins, or they're metabolites of some sort?
One is a metabolite. The other one is a very specific lipid.
Okay.
But we can imagine that in other situations, it's going to be a gene expression change. Okay. It could be a cellular phenotype that we can measure. But it's just, those are the first ones that we've identified.
So I'm going to come back to, how do regulators think about those biomarkers when it comes to patient selection in a minute, but not yet. Not yet. All right, Michelle, go to you.
So I think that there are many different exciting ways from a development and a regulatory strategy to think about novel modalities. Maybe just to answer this question, I'm going to go back to a personal anecdote from 3.5 years ago when my son, who has Duchenne muscular dystrophy, was diagnosed. He was diagnosed, and the first thing I did was look to see, are there clinical trials that he can participate in? And the answer was he was eligible for exactly 0, right? So for any parent, this could be disturbing. for somebody who spent their career in drug development, this was completely unacceptable. So imagine instead of having a situation where you go from not having a clinical trial available to saying, okay, this is this particular mutation. What clinical trials are being done to address this particular mutation? And doesn't matter what disease you have, you can still enter into that clinical trial. This is a possible strategy. So, and it exists already. They're called basket trials. And it's a commonly used practice in the oncology space. We're now starting to see basket trials being used in the rare disease setting as well. This is where you have clinical trials that have patients with a definition of a very different disease, but they all have that common mutation across the board. They're given one therapeutic and they measure the same or a similar outcome. And this has led to the approval of at least half a dozen different medicines already today. So, and our engineered tRNAs, we believe, are uniquely suited to leverage this basket trial strategy. So it's a many diseases at a time. And so instead of having a diagnosis where you go and you'd see what clinical trials might be possible, in today's world, where you may have no clinical trials, if we can use these basket trials in order to be able to capture patients across many different diseases, especially even ultra-rare diseases that wouldn't otherwise have the focus of a biotech company or a pharma company for a discrete innovation for that disease, this may really open up new opportunities and paths for those patients.
And have you had a chance to interact with regulators on this question? Because it's one of those things that sounds obvious in concept, but practically it sounds like it might be difficult. So how do regulators think about this type of approach?
Well, when you look at the novelty of clinical trial designs in the last 5 years, we see a massive uptick in basket trials like the one I described, or umbrella trials, which look at patients with different types of one disease, but different types of mutation, and depending on which type of mutation, you get a different therapeutic for each one, or a platform trial. These are becoming much more prolific. So the health authorities are becoming much more familiar and much more accustomed, and I'm really thrilled to see that they're becoming much more active. And yes, we are hearing a narrative from health authorities about many diseases at a time strategy is the only way we're going to tackle this challenge of rare and ultra-rare diseases, and are almost imploring the industry to try to figure this out.
I think there's really no other option if we want to sort of treat many of these rare diseases within our lifetimes, because there's just too many of them. All right, so I want to go back to the question of how do regulators think about, and how do you think about taking these precise definitions of patients based upon molecular profiles, and how does that lead to a clinical trial? Do you do a CLIA-approved diagnostic? Do you do all comers and then subset? Like, how are you thinking about this?
Yeah, well, so the way I'm currently thinking about it is that we're starting with, and we've chosen this, is to start with biomarkers which have some clinical precedentedness, some biological precedentedness, and there may actually even be an assay out there to measure it. So that decreases the activation energy to us. And so what we've done in engaging with KOLs and with regulators is to first gauge how willing are they to do trials like this. And to my delight, it's turned out that, exactly as Michelle's saying, regulators and KOLs are imploring us to do studies of just this sort that would allow them to characterize their patients better, choose patients for clinical trials in a specific way with a biomarker that we've either identified ourselves or there's an assay for out there already. And that the combination of clinical and biomarker characteristics will constitute the inclusion criteria for the trial. And regulators have been very, very open to that suggestion. Of course, what they want us to do, we're constantly going to them and saying, well, how good does the classification have to be? And you know what the answer is. They say, well, why don't you do the characterization, bring it to us, and we'll tell you whether it's good enough.
We'll tell you after the fact.
Which is
does
fair.
Create some challenges.
Right. And so Well, we're planning on starting not with all-comer trials, but with much more specific trials for a very practical reason. That is that these doing an all-comers trial in a heterogeneous population where one might have 4 or 5 or 10 different mechanisms requires a size and length of a clinical trial, which is often just not doable. It's certainly not doable for a small company, even for a big company. And so we have, in order to make the trials better, faster, cheaper, smaller, and shorter, we have to go to the segmented population. It has been interesting to watch what some of the companies who have tried to do this have seen with the FDA. FDA in some cases has actually said, well, gosh, I love your patient population, but you actually have to do a trial in all comers in order to convince us that your group is really different from the all comers. Yeah. And that we need to work on, because if we have to do the all-comer trial, that vitiates the whole reason of doing it in the first place. And I would argue is frankly not in the best interest of patients.
Yeah, it's going to be an evolving landscape over the next few years. All right, so we have about 5 minutes left, and I'm going to change the topic completely to sports. No. Who's going to win the national championship tonight? No, Jason. Platforms.
All right.
How do you think about building a platform? And as your programs advance, how do you think about investing in platform versus the product? And how does that evolve?
Well, you've actually spoken to this concept of an innovation stack, Robert, and I love that, so I'm going to steal that if that's all right.
I stole it from somebody else, so you're welcome to steal it from me.
Well, all good ideas. So the idea here is it's not a singular, and I suspect my colleagues will be able to say the same thing here. It's not a singular one invention. You know, if you think about drug discovery, the process of starting with patients and then going all the way, having an approved drug, there's so many different steps involved, and what you actually need is this stack of different technologies and inventions and innovations. And just to use Empress as an example, the way we thought about it is we built this data set. It starts with data. We built this data set of 30,000 individuals around the world. We built this data set of other species as well to look at. Then we use, I feel obligated to use the word AI. We do use AI techniques like natural language processing to interpret that genetic data. There's other computational algorithms and approaches. There's synthetic biology that allows us to generate compounds from that genetic data. There's mass spec and analytical chemistry technologies. There's other ways that we predict targets. So you sort of need innovations along that entire stack of technologies that adds up to, at the end of the day, giving you improvements from the start all the way through to a drug product.
Yeah. So. But as you build this, you spend all this time building this amazing platform in the stack. Then you start to make products, and then you need to fund your clinical trials. So what happens to the platform when you have to start to fund the clinical trial? How are you going to think about that balance? I know you're not quite there yet, but it's a challenge.
We are thinking about that because we do intend to be in the clinic quite soon. But I think it's people often ask this concept of platform versus products, and I think it's platform and products because if you're doing it right, what's happened is a true technology platform gets faster and better. So every time you do it, it gets a little bit cheaper, it gets a little bit faster. So the next product comes along a little bit quicker with a little bit higher probability of success. So I think you have to invest in both constantly.
Yeah. So quick answers from either of you. How do you think about platform product and making sure you invest in both?
Yeah, I mean, to me, it's really all about you need to be able to have a mechanism in which you can test, learn, iterate, and continuously improve, right? And you can't really do that with an individual drug. You've got to be able to do that with a platform. And that's how we continue to be able to, demonstrate, the power of our platform comes from the demonstration of clinical proof of concept of the assets that come out of the platform. But you can't make better assets without the platform, right?
I think that's a great
and I would just add that platforms allow you to learn, and learning requires n greater than 1. And so you have to, but the challenge for us and all of us is not just developing programs, multiple programs that allow us to test our platform, but to have each program be chosen very specifically to be generalizable, to say it's going to apply to the general case. So we don't take on every possible disease we could work on. Those diseases have to illuminate the general case so that we can take what we learn in example 1 and apply it to example 2.
Yeah.
But what we're also doing, and one of the reasons I'm a platform guy, I always have been, but one of the reasons that I don't like the term platform is it connotes something that you build once, like a deck on the back of your house, and then you're done.
Yeah.
And that is completely incorrect, of course, if you do platforms well.
Yeah.
It's constantly evolving based on what you learned from the last time. So you're constantly renovating the platform as you develop programs.
Yeah, so I think it's an interesting theme. You do need to build it. You need to show that it works with clinical proof of concept.
Yes.
But you need to build in the thought of whatever you show that works, you're going to need to loop that back to continue to evolve and build a platform.
Exactly.
So if you don't think about how your clinical study is going to help you build the platform, I think you've lost a really unique opportunity here. All right, so we're basically out of time. Last 30 seconds each, if you were to say one thing about JPM that you're excited about? Anyone?
Well, I mean, there's a lot going on in the world. I think you saw some examples here, seeing a lot of reason for optimism. There's a lot of innovative new approaches and technologies coming on board for patients who really need it.
Yeah,
So
that's
I'm
right.
optimistic about that.
That's good.
Yeah. To me, JP Morgan's all about collaborations and partnerships. How do we do what we're doing even better by harnessing the power of the broader ecosystem?
Yeah, that's great. So optimism, networking, and?
Yeah, and I want people, not only people who are JPM, but the larger community to know what we know and be optimistic in the same way that we know. We frequently, especially in our society, we tend to think, well, it's got to be too good to be true. It couldn't actually work the way people say it does. But I think what you're hearing was each of us have demonstrated that it actually can work to deliver for patients in a way we've never had the opportunity before. And so, but the only I would add about the partnering side is that each of us runs a very small company. And so we think very carefully about what do we do better than anybody in the world, and only do those things and partner for everything else that someone else can do as well or better than we can.
Yeah.
And that's what is so critical to this confab we call JPM, because that opportunity exists here to identify those people.
Yeah, so it's optimism, it's networking, and then being very smart about, I think, strategic partnering.
Yeah.
All right, so I think we're out of time. So thank you all very much for joining us, both in the room and also on screen, camera, outside of the studio. Clearly, I don't do this a whole lot. The metaverse, whatever it is. But hopefully we've convinced you that there are these advances in new technologies that are allowing us to think about going from ultra rare to more common, but then common to subsets of patients, which at the end of the day, it's really all about making a difference for patients. So I think we're on a good path to doing that. So thank you all for listening.
Thank you, Robert.
Thank you.